talk about some work that I'm doing in Singapore, and it's like more geared like towards biomarker development and using like novel biomarkers like for aging. And obviously the question is, we already have like a bunch of different like biological aging clocks, such as like the methylation clocks that are popular, transcriptomic clocks, proteomic clocks. And now I come here and say, well, maybe you should like develop a lipid clock. And I would argue, yes, we need like an additional clock in a way, because a lot of those clocks are still like experimental, and we currently don't know which one is the best clock. And a lot of them probably will like be like augmentative to each other. And as with normal clocks, certain clocks work at certain times of the day better, or like under certain circumstances. Like, I mean, if you don't have electricity anymore and only the sun, maybe like a sun clock still does fairly fine compared like to like a digital clock. So why lipids? One thing is like that lipids composition changes with age. We know that, and also we can like modulate lipids through diet and drugs and other interventions directly. And furthermore, lipids compared like to like certain other biomarkers are like already measured in the clinic regularly in every like basic blood panel. But for the people who are like not that familiar with what lipids are here, like just like a few comments about it. I think what is very interesting to begin with is like that we actually currently don't know how many lipids are out there, compared like we know how many genes are out there, and we kind of know how many proteins are out there. If you look at it, like proteins is like just like the second order derivative of like the genetic code plus like a few post modifications. Whereas with lipids, you enter like this very huge combinatorial space, which like almost gives like these explosions of like small molecules. And that leads to that lipids are like a very diverse and heterogeneous set of small molecules. They're usually like categorized in 8 categories. They have more than 80 main classes, 300 subclasses. And all the lipid scientists, they are adding more and more classes as they go along. They literally find like new classes that we didn't know before. And a few popular lipids that we know of are like cholesterol, testosterone, the hormones, estrogen, or fatty acids, bile acids. So it's like this huge category of like molecules that we commonly work in with the clinic or in science, but some people don't directly think they are actually lipids and just think of them as molecules in general. And what they have in common is they're like mostly hydrophobic or amphiphilic, so they don't like water that much. And furthermore, there's like a lot of variation between species. So like certain archaea bacteria have lipids that we humans don't have, and the other way around. But in general, there's like also like a lot of conservation around lipids. So like certain lipid species are like known in any kind of like living being, like from trees to bacteria to mammals. So what's actually the role of lipids, or the role of lipids in aging in general? Lipids are like, they're like for signaling, structural, and like energy storage. I think for like modulating aging, signaling can be like very powerful. If you think like steroid hormones, like that hormone replacement therapies and the like, increase like vitality, at least they make you stronger as in older age, and help you build more muscle mass. At the same time, estrogen seems to be useful like for perimenopausal or menopausal women to keep their health longer. And they seem to come with like some downsides though. But other signaling lipids are like very common all over the body, are like deacylglycerols and PIPs and prostaglandins in the immune system, and sphingosine 1-phosphate. They're like building blocks like for a lot of other signaling cascades. Then at the same time, you have like structural lipids that like give you strength, or like the structural components of like your cell membranes of your cells. And I always think they like might be very useful for measuring aging. And those are like your ceramides, sphingomyelins, glycerophospholipids, cholesterols, cardiolipids, and your mitochondria and the like. And then I think what people mostly think about lipids are like those lipids that are like energy storage, right? That actually the fat that you can see, like those fatty acids and triacylglycerols. But what I've been working on is like, can we actually use all these lipids in our body to estimate biological age? So use the lipids to predict how old is your biological age, or how good is your health actually. And we started that with like a proof of principle in worms. And what we did, we used like 4 different worm strains, like a normal aging strain that usually gets like up to an age of 30 days, 2 slow aging strains, which one has like a mutation, and the other one is like also a mutation on a mechanism related to caloric restriction. And then we also had like a strain that was like aging very fastly because it has like additional oxidative damage. We got like those worms at time points 3, 5, 10, 15, 20, so like over a longer time period, and collected in totally 168 lipid species, from ceramides to phosphatidylglycerols, sphingomyelins, like all the different classes. And then if you already look at just like do a principal component analysis of the lipid profile, you can already see that that down here you see at day 3 all the worms kind of start around at a similar age, or like a similar cluster. But as time progresses, you go up here and you actually find that like the day 10 wild type worms cluster together with the day 15, day 20 slow aging worms, which kind of indicates that the slow aging worms at day 50 and 20 are like more similar like to the day 10 wild type worms than like to the older wild type worms. So this was like a good indication that there like seemed to be like some age-related pattern in the lipid data. And if you also look at the principal components of the data, we found like that which is quite common, that the principal component 2 and the 1 and 3 are like fairly correlated with age. In the literature, that's what a lot of other clocks, speed methylation clocks, also find, that usually the first few principal components actually capture like the aging dimension in a way. It's also interesting to see that the principal components actually capture the different kind of mutants. So if you see here, like at the principal component 6, it's like fairly similar between the wild type and the slow aging ones, but it's like very different for the fast aging ones. Here, the principal component 7, it's like more similarity between like the wild type and the E2 and the MEF1, but this slow aging one has like entire different value, and here's the same like for a wild type. So which is cool to see. So now how do we build like this lipid clock? In principle, it doesn't matter if you use like the principal component analysis and it's followed by like elastic net regression, and then the idea is predict the chronological age of the worms given like this data. And what we do here is we actually use all the wild type data and the day 3 mutants, and the idea behind this is that like if we include the very young mutants, we probably already capture some of the changes that are only due to mutant status and actually not due to aging. And then we actually tested only like on the old mutants from day 5 to day 20. So those old mutants were never like in the training set. The algorithm actually doesn't know about them. We only use them for evaluation. And if we evaluate it, we actually get like a mean absolute error of 1.5 days. So in average, like the prediction is like within like 2 days of the chronological age of the worms. If we look at that, you see here like the black line is the wild type, and that's like a perfect fit, of course, because it was like in the training data, right? But if you look at the red line, the green line, and the blue line, all those red dots here were not in the training data, and they are actually like from the fast aging strain, and it's all above like the black line, which means they're aging faster, which makes sense. Obviously, it's a fast aging strain, we kind of know that, but also the clock predicts it. And what we see is like it's like roughly double the aging rate in the wild types. And the bottom ones are like the 2 slow aging strains, which is like roughly aging at half the age of the wild types, which also makes sense. Furthermore, what we did is like a Monte Carlo simulation where you see like the rugged lines here is like the real survival data of worms. And we used those aging rates that we found here and used them to simulate like Gompert's Markheim laws of mortality prediction, and those are like the thin lines. And what you can see is like the lines fairly well align with the simulations, which is like a good sign that there's like a useful signal in there. That's the same slide again in larger. But then obviously the question is like what lipids are like affected, what is like selected by the lipid clock. And what is interesting, if you look at like those first few principal components, you get like more of a signal. And what we usually find that like those LPCs and ceramides get like a lot of high values, or like a lot of emphasis on like the first two principal components, which like cover a lot of aging, whereas like in the principal component, the LPCs and the ceramides are actually both decreasing, versus in the principal component 2, which is like the one that is like correlated with aging for each of the strains. The ceramides are like increasing, but the LPCs are actually like decreasing, which is like interesting to see. So obviously, like worms is like good, like to have like this in preclinical studies, like in model animals. I think in worms is also cool because worms don't have methylation, so you cannot use those methylation clocks to estimate biological age. But every organism has lipids, which makes it useful. But obviously, ultimately we want to use it like in humans. So does this also work in mammals? And so we tried a similar concept out in mice, in the aging mouse brain, where we have like 64 different kind of mice. For each of those mice, we have like lipid data of 10 different brain regions, from the basal ganglia to the olfactory bulb, cerebral cortex, and the like, at age week 3, 16, 59, and 92. So from young to old. And in total, there are like 800 lipids in there. And we can build a clock on that. But for example, if you just like observe it first, if you look at the clustering, what you can see is like that like on the right side here, you can see that the different ages cluster well. You have here the 92-week-old one mice, here the 3-week, and here's like week 16 and 59. And also, if you look at the brain region, you also can see that each of those different age clusters has like different brain regions in them. So they also cluster in here. And what is interesting here, those 2 points is like the olfactory bulb, and they're a little bit outside of the other clusters. And what is interesting, the olfactory bulb is like one of those brain regions which actually has like neurogenesis, so that gets like, or creates like new neurons during a lifetime. So what we then find is like, if you look at like the lipid age for different brain tissues at week 92, is like that the cerebral cortex is actually like has like an accelerated age. So all those here should be 92 of chronological age, but you can see that like the mean of the median of the cerebral cortex is like more at like 96, and the hippocampus, the hypothalamus, and the olfactory bulb are like all more like around 90. So like they are predicted like to be younger. And what we also see is like there's like sex differences between the hypothalamus and the olfactory bulb, where like the hypothalamus is like the young hypothalamus, the female hypothalamus is younger than the male one, and the other way around for the olfactory bulb. So we also tried it out in humans, where we used like a data set of like that had like 70 healthy people with like a normal BMI, and 80 overweight people and 40 obese people in there. And we trained the algorithm on the healthy people, and then try to predict the lipid age on the overweight and the obese people. And what you can see is like that we have an accelerated lipid age for like the people that are overweight by 4 years, and for 6.6 in obese people. And that's also like what like methylation clocks in the like predict, that like if you're overweight or obese, your biological age is older than if you like just like normal, like within like the normal and healthy weight. And it's also like it's interesting to see this, like as a lot of other clocks, it has like this lipid clock has like a linear relationship with age, which we want to see. So advantages and disadvantages of those lipid clocks. I mean, some of the advantages I think is like that lipids are already part of the clinical biomarker panel, which might make it like more easy to get into the clinic, because clinicians already know about lipids. But like methylation clocks is like something very new and very novel, which might be hard like to get into the clinic. Then there's like a direct connection to underlying biology and disease that we can estimate and learn about it. Lipids can be fastly manipulated through diet and pharmacological interventions, so we can directly see if they work. Lipids react to changes faster, which makes it possible like to analyze interventions directly. And all species have lipids, like not only so it can be used preclinical and in the clinical space. versus like a lot of other clocks are probably can be only useful for like mammals. Lipidomics is like still like a rather underdeveloped and to some extent new field. It has its disadvantages that there are like no standardized panels and platforms. So like a lot of labs or measurements like might differ a little bit, which is like not ideal. And we also don't know how many lipids are out there and which of them are like very relevant for aging. We learn more about that, and we know that like certain lipids for sure are important for diseases. And furthermore, there are like not that many large scale data sets like to build like lipid clocks like that cover different ethnicities, different diets at different regions of the world. So that's something we also have to work on. So in summary, we can use the lipidome to estimate biological age in different tissues and organisms. Some of the results are like matching with epigenetic clocks. So it's somewhat of a validation. In all of the clocks, we see that like, ceramides having like very high and strong values. And ceramides are reported like to be changing with age. And I think that potentially there could be like a faster road to clinical applications for those lipid clocks if they work well, compared like to like other clocks. And yeah, with that, I want to acknowledge like my collaborators, like Jan in his lab, the lab of Marcus Weng, who did all the lipids, and then my supervisors, Morten Skybik Knudsen in Copenhagen and Brian Kennedy, who just spoke earlier, like for like the support they gave me and interesting discussions and guidance in the realm of science. And with that, I'm finishing up. And if you have like any questions or any lipid data, feel free to reach out to me. Thank you.